Okay, good morning, good afternoon, everyone. Welcome to the OTS 2022 webinar series. So we started this series last month by a webinar of the OTS president, David Geary. And we will continue these webinars with 2 webinars per month. So we feature one big name every month with a broader topic, and the other topic will be a presentation by 2 young Ph.D. students or early career postdocs who will present about their work. So also to give the younger people the chance to give a presentation for a broad audience. These webinars are recorded, so you can watch them on the OTS website. You can go to the OTS page and then click the webinar series, and then you can view your favorite presenter. So thank you all for joining, and I will give the word to Hassan.
Thank you so much, Ronald, and thank you everyone for joining us today. So I have the pleasure to present today to you our speaker for this webinar, Dr. Richard Geary, who has completed his PhD at the University of Texas, the College of Pharmacy, and his bachelor's in biology from Texas A&M University. His research is focused on pharmacokinetics, metabolism, and clinical pharmacology, with heavy emphasis on biochemistry. He is currently the Executive Vice President and Chief Development Officer at Ionis Pharmaceuticals, and he specializes in antisense medicines, as well as overseeing preclinical and clinical development of these therapeutics. He's a prolific scientist with more than 40 investigational new drug applications and 5 successful antisense medicine that are late-stage development programs. We're very pleased to have him here today to discuss with us some of his work, and the floor is yours, Dr. Geary.
Right, well, thank you very much. I'm certainly honored to have the opportunity to speak with you today and hopefully answer some of your questions. This should be a two-way, but as it will start, it'll be a little bit more one-way, with me sharing with you some of the work that we're doing with the antisense program. And so I'm going to share some slides. Hopefully this is going to come across. And like to discuss with you, in particular, chemical innovation that has been integral to the progress and ultimate delivery of RNA-targeted technologies. And so hopefully you can see and hear me and see the slide as well. Okay, so to get started, it's all about the chemistry. And on this slide, just kind of rotate around the modifications that have been studied. You know, it seems like every time I update this slide and then I present it, I'm behind. So this is a little behind, but it does kind of give a good snapshot of the various modifications that have gone into making RNA and DNA giving them medicinal properties to be able to develop these drugs. So the conjugates we're going to talk a bit about today. Of course, the GalNAc conjugate is represented prolifically in both the siRNA and antisense world. The sugar modifications also, you know, vary depending on the modality. Base modifications, not as much. In fact, over the years, while we've incorporated one base modification in our oligos, the methyl cytosine, which reduces the pro-inflammatory properties of the molecule, is really the only modification that has made it into the clinic on basis. If I could be wrong in some of the other areas, but certainly at Ionis and for antisense here, that's the only modification. So the real focus that I hope to bring to you today is happening here on the left-hand bottom side looking at phosphate modifications or base, what we would call the backbone of the oligonucleotide. More recently, one that's not shown here is the mesyl modification that has recently been published. We'll talk a little bit about that, but most of the work that I'm doing in the clinic has to do with the conjugates and the newer a few of the newer modifications and conjugates that are coming forward, mostly targeting transferrin 1. Okay, so I think for GalNAc, most are pretty comfortable and know a bit about this modality. The N-acetylgalactosamine or the GalNAc conjugated ASOs, they're modified on the 5' end with this linker and the GalNAc cluster of 3 N-acetylgalactosamine sugars. The cartoon on the left then shows how these enter the cell, excuse me, and ultimately for ASOs drive RNase H primarily in the nucleus, but also in the cytoplasm. And finally, on the right-hand side, you see 5 molecules that have now, 5 or 6, that have now been GalNAc modified. And these are clinical data over here on the right-hand side, showing the shift in potency. And of course, the shift in potency then drives a shift in safety. And I think about 15 years ago, I initiated a program, myself and our CEO at the time initiated a program to develop a structured database for which all of our safety data could be included once studies were cleaned and finalized. And that is used then to really drive down on the safety of these molecules. We now have more than 5,000 subjects administered with the GalNAc technology. Clinically, over 1,000 of these have been on the drug for 6 months or longer, a growing number at a year or longer. And the safety and tolerability profile observed in the early clinical studies support further development. And we've published that, and we're on the verge of publishing the phase 2 experience, which extends this out to a year. Safety margins are improved. We're really excited about the program. We have 16 of these molecules in development today. 4 of these have entered phase 3 registration trials. So obviously progressing the science and progressing these medicines to treat patients in need. So this is our cardiovascular snapshot, pinwheel, if you will, a pipeline that shows how involved we are in addressing cardiovascular disease risk factors. And I would say that it rivals some of the strongest franchises out there across certainly biotech, but also even large pharma. And what you see here is we're tackling a lot of lipids, hypertension, and TTR amyloidosis, as well as thrombosis. So I'm not going to go into detail here, but the reason I bring this particular franchise forward is kind of the focus. Every one of those molecules are GalNAc conjugated. So isn't that interesting? Every one of our cardiovascular pipeline now has been converted to GalNAc, essentially targeting the liver to target the heart. So you go after the liver where these targets are expressed and then in circulation, causing problems for the heart. So a little bit about PCSK9 here. I just show some of the recent data that was presented by our colleagues at AstraZeneca. in phase 1, they were demonstrating greater than 70% reduction in LDL, which now actually exceeds the activity of the antibodies, which drive about 60% reduction in the at steady state. This is after a single injection. And one siRNA currently approved, inclisiran, which has at steady state about a 50-55% reduction in LDL. So we believe we have an opportunity here to be a best-in-class molecule. How is this advancing? This is also called Ion 449, so it has to have its number nomenclature. And so we're currently potentially working towards a phase 3 program. We have shown in the phase 2B that this holds up, and with multiple dosing in patients with on aggressive statin therapy. So the continued development is supported by a strong database, and 2B data will be presented in April of this year at ACC, again, by our colleagues at AZ. This looks at a little bit of a phase 2, or actually, I think this is the multiple ascending dose in patients with on statins. And you can see that the PCSK9 levels are driven down to greater than 90% reduction with a once a month 90 milligram dose. And you can see how durable the effects are, which support not only once monthly, but possibly even less frequent dosing. The same is true for eplontersen. Now, this is our TTR LICA. By the way, LICA, which I use rather freely because it's part of our lexicon here, simply means ligand conjugated antisense. So LICA is really an acronym for anything that you attach to the molecule that aids in its delivery. So this is the dose escalation phase 1 data. We had greater than 94% at its maximum reduction of TTR, and this moved us directly into a phase 3 program. Just showing you some snapshots of some of the work that we're doing. These programs are well along, fully enrolled on the Neuro T-transform, and very close to fully loaded up on the Cardio T-transform. And you can see the designs where we're treating patients in polyneuropathy because we have a drug already on the market, Tegsedi, in this patient population. We're actually utilizing a rather unique pathway of a historical placebo group from our previous study. And we'll be using the data from this study and a comparator group with Tegsedi or inotersen there, which was the first generation without the GalNAc, and this is now the second generation with the GalNAc. And then that moves into long-term. Everybody gets the GalNAc after 35 weeks. So we're approaching the 35-week interim analysis time point this year. And we're pretty excited to be able to bring some data from this program, which should actually also have some read-through to the Cardio T-transform study. So these phase 3 studies are underway. And what I just wanted to hopefully bring home to you is the research that we're doing in cardiovascular disease, cardiomyopathy, heart failure, atherosclerosis, you name it. We're looking at it and trying to tackle it with multiple targets, which is a principle for building a strategic pipeline. And it's our number one principle when it comes to complex disease, and that is we go after multiple targets. The RNA targeting, of course, uniquely positions the industry to tackle this goal. And of course, the strategic goal here is to ultimately provide multiple tools in the cardiologist's toolbox with one goal, and that is to save lives and to actually change the course of these deadly diseases. Cardiovascular is still number one in terms of fatalities. So it's certainly the arena we are focused on. Well, what about the CNS? Want to move a little bit to the brain and talk to you about some of the issues, challenges, opportunities associated with targeting the brain. Of course, we have a neurology franchise that's quite large, partnered with both Biogen and Roche, as well as our own programs, which are now in the clinic, and a couple additional ones starting this year. So in ALS, I just want to draw your attention that we have 4 different targets going after ALS or Lou Gehrig's disease. It's a very rapidly progressing and horrible disease. We have both the genetic forms, which are looking at SOD1, C9, and FUS in the clinic today. In fact, FUS and SOD1 are in phase 3 programs, while C9 is just completing its phase 1/2 program, and Ataxin-2 is just initiated. But all of these are in the clinic today, and they tackle pretty much all the forms of ALS. So we feel like we have ALS kind of surrounded, and hopefully we're going to be able to make a real difference there. So it's an exciting time. I want to talk to you a little bit about FUS because there is some new news that has just come out recently and is in fact on the OTS website. This is the 3rd most common genetic form of ALS. It's very rare, about 25% incidence compared to the SOD1 ALS. It's fast progressing. It's pretty terrible. It actually takes the lives of young people, and that's where the whole program got started. Dr. Schneider had a patient, excuse me, that is described in the paper that's on the website at this point. And what you see in that paper was the use of ion 363, which silences the FUS expression, did decrease FUS pathology and reversed insolubility of some of these particles that accumulate in the neurons and ultimately result in death of neurons. And so we were excited when we started seeing the data from this. It became pretty clear that there were a number of patients who were coming forward and wanting access to the drug. And so in our discussions with the regulators, we decided to go ahead and initiate a phase 3 program. This, I think, is a pretty good representation of what we're about. We don't just do big diseases. We also do very rare, and in fact, I would call this nano rare disease. There aren't a ton of patients. This study will enroll a little over 60 patients, both juvenile and adult patients. The first part of this study is controlled with placebo. It has an escape mechanism for people who get into real trouble. Following a certain period of time, 29 weeks, then all patients are brought onto the product, including the placebo patients. this is really, this kind of a design is not necessarily unique, but there aren't a lot of designs like this where a part of the program that develops the data that will ultimately drive the approval is actually being collected in open label extension. So excited that we now have a number of these patients on the trial, and they are coming out of the woodwork. What I wanted to focus on a little bit today was kind of the challenges and opportunities in the research. What I'm showing here, of course, is that the drug, when it's given subcutaneously or IV, does not cross the blood-brain barrier. And so we aren't able to attack diseases of the brain with systemic administration, but rather all of these drugs that are in our neuro pipeline are administered, with the exception of one that is targeting a molecule in the liver, or a target in the liver. With that one exception, all of these are administered intrathecally into the space of the spinal cord, into the cerebrospinal fluid. That results in very nice distribution. And the research team here have developed a very complete compendium and atlas of the brain from our research in primarily rats and monkeys, and now starting to expand that to some of our clinical experience and utilizing even radiolabel non-invasive techniques to really understand distribution in humans. So it's all about distribution, but the route of administration is a challenge, you can imagine. And so we don't want to have to give really frequent intrathecal administration in diseases that maybe aren't as severe as a rapidly progressing class ALS, which will require relatively frequent administration. But we'd really like to get to the point where we're administering the product into the cerebrospinal fluid, perhaps even once a year. And in fact, Biogen licensed our technology, our first technology push in a new chemistry that does just that. Based on our preclinical work, we have a molecule that has an extended half-life with a new chemistry targeting SMN for as a follow-on essentially to Spinraza. And so that is our first and primary goal, which has been running kind of in parallel with another goal, which is perhaps a little bit further out, but equally a challenge and opportunity, and that is trying to deliver drugs across the blood-brain barrier, these particular oligonucleotides across the blood-brain barrier. Today, we don't have that, but we're moving forward with technology that might get us there. And so in parallel, we're looking at bicycle technology as well as other technologies, even antibody technologies that target transferrin 1. And everybody, I think, is probably fairly familiar with this, but may not be as familiar with the bicycle technology. Bicycle is able to take diverse phage libraries, very large phage libraries, and optimize leads on particular focused targets. In this case, transferrin 1. These are then transformed into these loop scaffolds that target the binding, and it's relatively small molecules that are then conjugated to ASOs. We've shown that we can conjugate these to ASOs and drive really nice biology in muscle as well as heart, and we're beginning to look then at exploring the potential use in regard to crossing that blood-brain barrier that is such a formidable barrier to achieving distribution in the brain. Now, there are advantages, as you know, to not being able to go to the brain when we're targeting the liver. So it's very nice that we don't have to worry about off-target hits in the central nervous system. On the other hand, when we want to be in the brain, there is only one way to do it today, and so we're exploring the potential for the use of this technology to perhaps get us to that next step. So one point that I wanted to make on this slide is that at Ionis, we really, in our heart of hearts, believe it's really important to publish. Maybe it's a little bit easier once you get into the clinic, because a lot of what is going into the clinic is already public, and there isn't a lot of proprietary information built into these programs. But I just want to make sure that the Oligonucleotide Therapeutics Society is always focused on sharing data and getting data out as soon as we can. Obviously, in industry, we have proprietary components to our research. Nevertheless, as soon as we can publish it and it's protected, we should be getting it out there and being able to share with the broader. And this just shows some of the publications over the last, I don't know, 5 to 10 years in the New England Journal of Medicine and the Lancet. So very broad publications on our clinical development. We also have published our database, I talked about a little bit ago, our safety database in the Nucleic Acid Therapeutics Journal. And I think it's really important for us to continue to share what we're learning as we go forward with new technologies and in essence, new pipelines like the GalNAc pipeline. And we published the phase 1 safety data in NAT, and then we're now following up with a phase 2 up to 1 year experience. And so I think ultimately my vision is to always be pushing the sharing, if you will, thresholds. So a little summary here before we go into kind of I think a Q&A time. RNA therapeutics have the potential to reach new targets, and many, if not most, should be the undruggable targets that aren't really available to other modalities. Certainly, the greatest need, the need is very great. There are many at-risk and hurting patients, and so focusing on patient-centric drug development is another passion of mine, and the vision of potentially reaching the heart and the mind, which we've looked at today, and beyond, is well on its way to realization. And I think innovation in delivery technologies are making significant progress. A lot of that, if not most, is in the chemistry, if you will, the chemistry of these molecules, paving the way to a lot of modalities. We are looking at multiple modalities. We're not just looking at what we can do to make antisense better, but also siRNA as well as CRISPR. So broadening our toolbox, at least on the research side of the house. So thank you very much for hanging with me through this little kind of walk through technology and some of the research that we're involved in, and a little bit of my vision also for the society, which is is really to continue to utilize that as a base for sharing and for developing young investigators. And that's part of what we're doing with these webinars, is getting this information out and making it public. So I'm very happy to take any questions at this time, and maybe make this a little more of a conversation and less of a didactic talk.
Yeah, thank you, Richard, for this very nice and clear overview. So for everyone, so if you have a question to Richard, so please use the chat function. So then Hassan and I will read the questions and ask questions to Richard. So I see that there are already quite some questions in the chat, but since I'm the host, I can start with my own question. So a question to you. So I think you've very nicely presented all kind of modalities, different antisense modules. So, but I think we have to go one step back. So how do you decide that the disease is eligible for your for antisense therapy, and what kind of diseases are eligible? Is it easy? Because what do you need to know? Because what I can imagine that if you work on a gain of function mutation, that this might be easy if you can just delete this particular protein that is causing the disease, and that might be very beneficial for the patient. So what kind of information do you need to work on a particular disease?
Yeah, so understanding if there's a genetic component and whether there's a kind of a single component that's driving the disease. That's probably number one, because these are very specific molecules. We're not generally hitting a large variety of pathways. So it's one pathway, and really understanding whether that can make a difference. So sometimes we don't know. I give the example of angiotensinogen. Angiotensinogen, of course, sits at the top of the RAS pathway. And in cardiovascular disease, you would say, well, if you hit that, you might do some real bad things to the kidney, because we already know that if you go after the RAS pathway and you're hitting that in the kidney, you would have a problem. So the question was really, if you hit the very top of the pathway, one, can you do anything with blood pressure? And two, do you have to hit it in the kidney, or are you able to go where the oligo goes, or could we put a GalNAc on it and do the same thing by targeting a hepatocyte and keeping it away from the kidney? That was all unknown, right? The only thing that was known is the RAS pathway is important. So we went after it, and we did a lot of the preclinical work to really drive whether, and it took a number of years to really understand whether this is a safe pathway and whether we can deliver this safely and make sure that we're not going to be hitting the kidney. So that's one, I think, example. There are multiple. I think any gene you can think of can be reduced with a gapmer, RNase H, antisense, or an siRNA in the liver, in the hepatocyte. If you can think of it, you can knock it down. The question is it going to make a difference in the disease? So a lot needs to be understood about the target, not just on the positive side, but also on the negative side. So how necessary is that target? Is knocking it down 95% a good thing or a bad thing? And so all of these things need to be taken into consideration. The number one, I guess, when you ask, well, what do you think of when you're thinking about which diseases to tackle? Well, number one is, can we get there, right? Where it's expressed. That's number one. And so, of course, the field has been replete in looking at the hepatocyte because everybody can get there. But then outside of the liver, kind of the frontier now. And of course, there are diseases that we could tackle in the muscle or even in the cardiomyocyte, but we don't easily get there. And so are there things then we can do with conjugates that get us there safely? And it's a whole nother frontier. So that's why I just tickled a little bit on the peptides and transferrin 1, because that really covers muscle, cardio, and some little bit potential for crossing the blood-brain barrier. But those are the things you think about. So delivery, number one. Number two, the learning about the target. Does it really play a major role in the disease? And sometimes we don't know until we get into the clinic. So there are cases where we have animal models, they're not perfect, where things are looking great, we get into the clinic and it's not the dominant pathway or whatever may happen. So it's still drug development.
Is there a difference if you think about so I think delivery, so reaching the target cells? I think that's easy. You know mostly which tissues are affected. But so is there a difference between very common diseases and the NS1 or the nano diseases where there are only a few cases? So do you need to know more about the mechanism working on common diseases if you compare this to the NS1 diseases? Or do you think it does not really matter? You need to know the mechanism anyway.
It's a great question. I think it really depends on the disease. I mean, some of the very rare diseases, it's very clear what the mechanism is, and it's monogenic. So a monogenic rare disease is clearly an area where you can work, and if it's gain of function, it's a no-brainer. Of course, there are ways to turn on genes. And Spinraza is actually a drug that increases production of a valuable protein, right, SMN. And so antisense can be used to either audit, cause a change in editing and improve the production of a protein as well. So you can have loss of function approaches. It's just not the majority of where we work or where the diseases are. Does that help?
Yeah, it's very clear. Yeah, well, I was looking at Hassan if he takes over the question, so it was very clear answer.
Okay.
Thank you so much, Richard. I have one question to you as well. What new risk you think, if any, that could arise when we start targeting the cardiovascular diseases specifically? Because that's kind of like a new area for us in nucleic acid therapeutics. So do you think there's like any new risks other than the ones that we know once we start looking at such targets?
Yeah, a lot of the targets are well known. There are certainly heart failure targets that require distribution beyond the liver. But for the liver targets, it's fairly well known. Now, what other things do we deal with when we get into some of these broad diseases? Well, one of them is you're going to be treating disease in millions and millions of patients. So all of a sudden, a small safety risk becomes big. So I think when you get into broad disease, two things, you're going to be moving into very large populations. And of course, there's all kinds of business risk and costs and those kinds of things associated with doing cardiovascular outcome trials. Most cardiovascular outcome trials, probably 50% or more, fail. So who has the wherewithal to tackle $1 billion clinical trial? There aren't a lot of companies that do. And so you need to choose probably targets that are fairly well understood and perhaps justified by epidemiology as well as preclinical model. It's not just a preclinical model kind of exercise. So I use the example of the LP(a), which now a lot of people are getting involved in. We're nearing full enrollment in a cardiovascular outcome trial for LP(a) with our partner Novartis. They had the big bucks and the big teams to run a cardiovascular outcome trial in these patients, but it was the epidemiology that drove the justification to actually spend to do that experiment. So we think of our, for me, a mouse experiment, I can do a few of those with very little business risk, but a very large outcome trial has a very large risk component. So anybody that's going into that area is looking at all kinds of things. So when we started pushing doses on buprenorphine, as an example of a drug that was in phase 2B, and our partner Pfizer decided, no, we're not going to phase 3. We're not going to go do that $1 billion study. They were looking at maybe something that's not at the dose you need, but it's out there, and any little risk, a company may not be willing to take that kind of risk. So there are a number of risks beyond the biology, I would just say, when you get into cardiovascular.
Thank you. And we're going to take a couple of questions from the chat. So a question from David Corey, and also in the same line from Miriam. They're asking that inclisiran seems to be a very strong and well-tolerated drug. So can you speak more about how does that compare to your anti-PCSK9 drug that you are developing?
Yeah, I think that's a great question, because inclisiran is a drug that's being marketed and delivered out to patients in a very different model. The model that we would be developing with AZ is more of a self-administered modality, probably more like the antibodies. It's certainly supporting once monthly, very safe. And so from that perspective, it's a fairly easy delivery to a patient. To have a patient self-deliver a quarterly or once every 6 months, it's probably not as easy, and probably properly being handled by the clinician. But I think from the perspective of potency, the drug at very low doses drives more than 70% reduction in LDL, which every little bit helps, right? We know that LDL linearly drives cardiovascular disease. The more you can control in a difficult to control patient, the better. I kind of compare it to Crestor, since that's the AZ story, right? And I'm sure they think about it as well. So Crestor came on as the 7th, 8th, 9th, 10th statin. Everybody was doing statins. There were good statins out there. Lipitor was fantastic, took over the market. Crestor even had some warts, and there were some concerns. But it was the most potent drug. You could give 5 milligrams Crestor and get the same you get with 20 milligrams of Lipitor. And so it became kind of this story of the newest, best in class, and Crestor eventually took over the market. So I think you've got, a situation here where there's a business component, and you probably need a better molecule. That was certainly part of the strategic role of the PCSK9 for AZ. was to drive to having the best in class. And then, you know, a large commercial organization believes they can win.
Thank you, Richard. So another two questions that are a little bit related from Supriya and Sylvia. They're asking if you've seen that the Morpholino or CAT-BNA modification increases nucleus stability. And then a follow-up from Sylvia, do you have any Morpholino-based oligos in the pipeline?
All right, so the second part of that's easy. No, we don't have any Morpholino. We can certainly practice Morpholino. The issue with the Morpholinos is they have poor drug properties in terms of distribution. So where if you give a Morpholino sub-Q or intramuscular or IV, most of the drug will end up in the urine very quickly. So it's not it's the druggable properties, the amount, if you can get the drug into the tissue, it's very stable and it works. But we feel like we have better chemistry.
Okay, moving on to other questions. So we have a question here saying that Ionis has been working on many impressive new chemistries to mitigate some toxicity. Have some of them been advanced to clinical trials in terms of the new chemistries?
Yeah, so the answer, of course, is yes. I showed you our entire database on the Downhacks, which have improved margins of safety. And of course, the methylcytosines are on all of our molecules. That was to reduce pro-inflammatory effects. There are additional modifications that are in the research stage that have not yet reached the clinic. The 2'-Mo chemistry was brought forward because the first generations had a lot of, not only the shorter half-life, but also a lot of off-target effects. We couldn't even give those molecules sub-Q. So the advent of 2'-Mo drove another level of safety. Again, the CET was another approach to delivering less drug with greater activity because of the greater affinity of binding to the target. And we could also work with shorter molecules. We've removed phosphorothioate from the backbone of most of our, not all phosphorothioate, but a significant portion of the phosphorothioate from most of our CNS drugs. And those are all in the clinic. So I think, yeah, there are a lot of, there's been a lot of progress on the chemistry, and there continues to be. Not all of these are yet in the clinic, but many are. Good question.
I'm gonna squeeze in two more interesting questions here. One from Josephus, who's asking if we will be seeing more chiral/stereo-random MSPA chemistries and something we're going to see in the future, basically, as ASO candidates.
You know, I couldn't tell you today which one, but yes, the answer is yes.
Okay. And another question from Pedro, who was saying it's a great talk. Could you comment on the necessity for future multi-targeting approaches, for example, using 2 or 3 or multi-ASOs to target concurrent pathways or genes in complex diseases?
Yeah, we know that that's possible. The regulatory hurdles are a bit more difficult, but not impossible. Certainly, oligos, and you know, we've looked at this in cardiovascular disease. You know, could you put together a cocktail of 2 or 3 of these molecules that pretty much takes care of everything? And we know it can be done from the bench we know we can put it into animals and it will hit all those genes. Determining what needs to be done from a safety perspective to get to that level is something that we're exploring. It's not the first time that something has been, there actually has been a clinical program, I believe it was an ophthalmology approach, where, and maybe an inhalation, also a pulmonary approach with multiple drugs, multiple antisense drugs delivered simultaneously. They're perfectly compatible as long as their chemistries don't bind to one another. If you get to two sequences that are sense and antisense, you might have a problem negating the effect of one or the other or both. But as long as you've got sequences that are independent, it's not a problem, particularly with the increased potency of these molecules. But you do still have to worry about obviously the safety of all of the combined chemistries, and so that's probably the main challenge, is developing an acceptable path.
Thank you. So it might also be a very good approach for many people. So I have another question. It's also related to a lot of specificity. So there are also targets that need allele specificity or an allele specific approach. So can you elaborate a little bit on this? And are you working on this, or would this be a good approach for Ionis?
Yeah, absolutely. We have, you may remember in the Huntington story, when we were working with Roche to determine which of the molecules to take forward into development, we had an allele specific and we had a non-allele specific. And the non-allele specific molecule was the one that was chosen to go forward, tominersen. The allele specific, yes, it can be done, has been done, and sits in the wings, depending on where the business takes us. But at this point, it hasn't been taken forward. We do have an allele specific product in the clinic today, targeting Rho, a Rho mutation in the eye that causes a type of blindness, very rare, but it's an allele specific because that target, the normal target, not mutated target, is needed for health of the eye, and that is a product that is being developed by one of our partners. So how is it done? So one of the ways that we've looked at doing allele specific targeting, and of course in Huntington's you have to remember that there are multiple mutations, right? And so if you go allele specific, you want to pick the one mutation that maybe is dominant, but that's still only a fraction of the population. And that was one of the reasons that Roche moved forward with the non-allele specific approach, is because we could target all patients, not just a particular mutation. Nevertheless, it certainly can be done, and using chemistries that are very potent and have high affinity, it has been the way that we do that, and we build the chemistry around the mutation so that the high affinity allows you to actually select your binding. Now you have to have, it's like single mismatch, you lose all your activity. And that's certainly what happens when you go after allele specific, is it has to be extremely specific, and a loss, it has to lose its function with a simple one base mismatch. So that can be done, and it has to be built in a certain and specific way. It's a great question.
Any idea how many molecules are needed to treat like the entire Huntington population? So can you treat more or less the entire population with 5 compounds, or do you need 10 or 20?
Yeah, it's kind of like when you think about DMD, right? Duchenne's, where you have multiple. I don't know the number, but it is, yeah, it would be multiple medicines.
The last question from the chat. So if it's a question from Ma, if I understood correctly, the TTR Neuro program also uses GalNAc conjugation. What is the motivation behind it?
So there were two things. So Specificity is on the market today with a black box warning for renal and platelet issues. So the primary goal was we have a target, works very well. Can we get out from under those safety issues and have a product that is better for the patient? So that was the driving force for looking at polyneuropathy with a GalNAc. The dose is about 1/30 of the Specificity dose. We've modified the molecule a bit. It's got the GalNAc on it, and it's extremely safe. We now have multiple years of experience with the product, and you're not going to see those problems with this molecule. So it was driven almost exclusively by safety, because the efficacy is tremendous for Specificity, and we believe that we'll match that or exceed it. Okay,
thank you. So these were the questions from the chat. So I have one more final question. So that's an OTS related question. So what would be your advice to young people that are searching for a career in the oligo field? So what would you give as an advice to young people?
So that is a great question, and probably takes more than a minute. But I would say get involved with a society where you can actually see the breadth of what's going on, find out where your passion is, and join in. And it may be in academia, where a lot of good stuff is happening, or it may be in industry. But you really want to get to know the field if this is something you're thinking you might want to be involved in. And so get involved. You know, this is a little self-serving, but get involved with OTS and really understand the full scope, because in OTS, you're going to see the whole field, and you're going to be able to interact with people that are involved in the research and have a passion that might match yours and might not.
I think this is a very nice and great answer to the question. So hereby I would like to close the webinar. So Richard, thank you very much for this excellent presentation and a nice coverage of all the questions. So we're very thankful. And I would like to say to the audience that the next OTS webinar will be on February 24th. And then Hassan Faki will present some of his work and some a vision about the junior trainee reps on the OTS. And there will be a second presentation by Chantal Ferguson about her work in the Corova lab. So please join this webinar February 24th. Thank you very much for watching the webinar, and I hope to see you in 2 weeks.
Thank you.
Thank you, everyone.